All right. Hi, everybody. This is our Friday review session for week 4. If at any point you can't hear me um, or something, if there's any like technical issue, please let me know. We'll try to fix that. If you have any questions, you can, of course, feel free to unmute your mic and ask, or you could drop it in the chat box. You could DM me privately or just write it like for the whole chat. That's totally fine. I'm going to try to be monitoring it the best I can, but we'll see how that goes. Okay, so let's do this. All right, so I know the majority of the questions based off like what I've heard from other TAs and what I had in my own help session. I know the majority of the questions are on the trip operon attenuation and RNA interference. So I'm just gonna kind of gloss over this, all this information about the RNA polymerase and transcription factors. But of course, like if you have questions, please let me know. But if I go fast through this section, it's only because I want to get to the trip operon as quickly as possible. So with that being said, I'm gonna go on. So there's the RNA polymerase and transcription factors. So the RNA polymerase, this giant thing over here, that's pretty much just responsible for doing the transcribing, right? Whereas transcription factors, those are proteins that help the polymerase do the things, like binding onto the, transcription factors, they bind to RNA polymerase, they help RNA polymerase bind to the DNA, and they do something really important. They either promote or suppress transcription. So some transcription factors, yes. Sorry, Said, I just got your text, but I don't really know what you're asking. Some transcription factors promote transcription, so that would be fine. Others suppress it. So if the cell doesn't want something to be transcribed, it would probably use a suppressing transcription factor for that. Yeah. Okay. There are two kinds of proteins. There are specific sequence, there's sequence specific and there's non-sequence specific. So as the name implies, the sequence specific binds to a specific part of the DNA, whereas the non-sequence specific binds to really any part of the DNA. And again, these are important. So let's look at these proteins over here and let's talk about, can you guys tell me, like, what do you think DNA polymerase is? Is it sequence specific or non-sequence specific?
Sequence specific.
Why would you say that?
I mean, I think it's just because like DNA polymerase just generally has to follow a specific sequence to transcribe nucleotides. Am I thinking the right thing?
Yeah. Sorry, I was just looking on my phone. Okay, I hear you. Yes, the DNA polymerase is responsible for transcribing like parts of the DNA. However, the DNA polymerase needs to be able to transcribe any part of the DNA, not just specific places. It needs to be able to transcribe the whole thing if it wanted to. So the DNA polymerase is actually a non-sequence specific. And I know that that sounds a little counterintuitive, but DNA polymerase is actually a non-sequence specific protein because although it almost always is guided to a specific place, that doesn't mean that it can only transcribe a specific sequence. Technically, it could transcribe the entire thing if it wanted to. Does that make sense?
Yeah.
Okay, cool. How about histones? What do you guys think for that? Histones, if you remember, those are those little ball proteins that DNA wraps around. Histones are responsible for essentially like closing the chromatin or opening the chromatin to allow for transcription. Do you guys have any guesses as to what histones might be? That's okay. Okay, so histones, also like DNA polymerase, are also non-sequence specific. That's because histones, like I said before, histones are like those protein balls the DNA wraps itself around. So those protein balls need to be able to, or I guess they are able to wrap itself around any part of the DNA. There's no specific part in the DNA that is like just for histones. it could really be anywhere. So histones are also non-sequence specific because you can really have histones at any part of the DNA, or they can bind and wrap themselves around any part of the DNA, not just a specific sequence. How about transcription factors? So in the chat, sequence specific, yes. Transcription factors are certainly sequence specific. Transcription factors, like we mentioned before in the previous slide, yeah. Transcription factors are responsible for grabbing the DNA polymerase and putting it at a specific spot. So like someone said before, even though the DNA polymerase does transcribe very specific parts of the DNA, it's not the RNA polymerase that is sequence specific. It's the transcription factors that take the DNA polymerase to a specific sequence. Does that make sense? Any questions so far about this? Okay. Okay, and just like there were two kinds of proteins, sequence specific, non-sequence specific, there are two kinds of transcription factors. So I kind of touched on this before. Oh, wait, no, I didn't. Sorry, anyway, there's general transcription factors. These are the ones that help RNA polymerase bind to the DNA. They have no effect on transcription. So again, transcription factors are important because they take the RNA polymerase to the DNA and they help bind it. And two, they can be responsible for promoting or suppressing subscription. And that is what a regulatory transcription factor is. Before I move on to regulatory transcription factors, I just want to give an example of a general transcription factor. Sigma factor 70 is a general transcription factor found in prokaryotes. So that's an example. Sigma factor 70 is the transcription factor that's responsible for putting the RNA polymerase onto bacterial DNA. Okay, regulatory transcription factors, these do, as the name implies, they do regulate the rates of transcription. These transcription factors come in two flavors, positive and negative. A positive regulatory transcription factor promotes it, so it allows the RNA polymerase to do its thing. Negative regulatory transcription factor, as the name implies, it suppresses transcription. So each of these positive and negative transcription factors, they only really work when they're bound to the DNA. So if they're not bound to the DNA, they're not really doing anything. Here, I just put a little side note, RNA polymerase. I don't know how important this is, but I might as well just like talk about it. The RNA polymerase lets go of the transcription factors once it starts. So after the general transcription factors put the RNA polymerase onto the DNA, and after the regulatory transcription factors are like, yeah, you're free to go, or no, you can't move yet, then all of those things disassemble and the RNA polymerase just does its thing on the DNA. Okay, any questions about this? I'm sorry if I'm going a little fast. Like I said, I just wanted to move on to what I think the bulk of the questions are, which is this stuff. Okay, so in prokaryotic DNA, there is something called a trip operon. There's a bunch of different operons, like the lac operon, whatever. Right now, we're only going to be concerned with the trip operon. So I'm going to talk about the anatomy of this in a second. So this is your DNA, right? You have your repressor region, which codes, this region actually also codes for the repressor protein, which I'll talk about what it does. The regulatory region is what the name implies. So remember, regulatory regulates the rates of transcription. So this region is just pretty much telling RNA polymerase, which again, I'll get into a lot of detail about the regulatory region. But this region is just responsible for, okay, is the RNA polymerase able to transcribe what it wants to, or does it need to suppress transcription? And these are just your structural genes, the genes that will be responsible for eventually synthesizing tryptophan. Okay, so what you need to get out of this level of regulation. What you need to get out of this is you need to consider two things, you need to consider two kinds of concentrations. So there's tryptophan concentration, and then there's tRNA tryptophan concentration. It would help a lot if you consider these things as two different things. So once again, there's a tryptophan concentration and there's a tRNA tryptophan concentration. And although they kind of go hand in hand, it really helps if you consider these, it really helps if you consider these as two different things. Okay, so when we talk about this level of regulation with the repressor protein and whatever, we're only considering the tryptophan concentration. Let's not worry about tRNA tryptophan yet. tRNA tryptophan is going to come in handy later. For now, let's just worry about tryp concentration. So when the tryptophan concentration is high, meaning that there's a bunch of tryptophan molecules like just diffusing around in the cell, they're just like la la la. If there's a high concentration of tryptophan floating around, it's likely that some of that tryptophan is going to bind to the repressor. So if tryptophan binds to the repressor, this thing, is going to bind to the DNA, which blocks, it's actually going to connect to here and here. It's going to connect to the regulatory region. Actually, this isn't like super important. What the hell just happened? Yeah, that's not too important, like where it binds. Just know that the repressor will bind to the DNA and it's going to block sigma 70. Remember, sigma 70 is your general transcription factor, which is responsible for putting the RNA polymerase onto DNA. So if your repressor is bound to DNA, it's going to block the sigma 70 site. If it blocks the sigma 70 site, the RNA polymerase can't get onto the DNA, no transcription will happen. Okay, so that's if tryptophan concentration is high. When tryptophan concentration is low, as you might have guessed, there's no tryptophan floating around in the cell. There's no tryptophan available to bind to the repressor, which means that the repressor does not bind to the DNA, which means that the RNA polymerase and all the transcription factors are able to bind to the DNA and start some kind of transcription. So this is like the first level of regulation. No, this is like the first level of regulation. Sorry, that was super dramatic. Oh, geez, I don't know what I've done now. I don't know what I've done now. I'm so sorry. Sweet. Okay. Can you guys still see the screen? Okay. I also can't see the chat box anymore, so I'm sorry if you're trying to say something.
Yeah, I can see
You're
it.
saying, yeah, it's fine.
Yes, we can see the screen.
Sweet. Okay. Okay. So does anybody have any questions about this level of regulation so far?
No.
Okay, good. So just to reiterate one more time, what you need to get out of this level of regulation is when tryptophan is high, the repressor is bound to DNA, no transcription will happen. If tryptophan levels are low, the repressor is not bound to DNA, and transcription is allowed to occur. Okay, so remember how I said we need to consider two different things, tryptophan concentration and tRNA tryptophan concentration. So when it's high, nothing is going to happen. When it's low, there's another level. Well, I'll get to that in a second. When tryptophan levels are low, the RNA polymerase is allowed to bind to the regulatory region. So let's look into the regulatory region a little closer. Everything that you see here, this whole diagram is just this part. It's just the leader and attenuator region. So again, this whole thing that I'm about to talk about in a second is just this stretch right here. So we know tryptophan levels are low because the RNA polymerase is allowed to bind to it. But now let's see, let's go into that a little further. Now we need to consider what is the concentration of tRNA tryptophan. So we know tryptophan concentration is low. Let's talk about when tRNA tryptophan is high. So when tryptophan level is low, the RNA polymerase is going to start transcribing. It's going to start here, somewhere around here. You can see my cursor, right? Can you see this okay?
Yeah.
Sweet. Okay, so the RNA polymerase is going to bind somewhere here, and it's going to start transcribing. It's going to go la, 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 all the way, blah, blah, blah. And it's going to pause right here. It's going to pause right here, which is like region 2. Right here where it says transcription pause, the RNA polymerase is going to pause. It pauses to allow the ribosome to catch up. So a little bit upstream here, you have your ribosome that's following the transcript, and it's going la, 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 and it's translating at the same time that it was being transcribed. Okay, so let's talk about when tRNA tryptophan is high. So if tRNA tryptophan is high, the ribosome is going to be able to translate through this region right here. Oh, geez. Mind you, the ribosome is translating all the way up to here. Once the ribosome gets to right here, to this region, it's going to ask itself, do I have the amino acids necessary? Do I have the tRNA necessary to continue translating? So if the case is yes, that tRNA transcription is high, then yeah, the ribosome is going to translate straight through this like it's no problem. So because tRNA tryptophan is in high concentration, the ribosome is going to continue translating. No big deal. It's just going to keep going until it gets to this stop codon in region 2. Okay, any questions so far? Okay, so once the ribosome gets to the stop codon, which is right here in region 2, right? The ribosome is going to stop. It's going to stop because it reached the stop codon. So it's going to pause there for a minute. Once it pauses there, the RNA polymerase is going to continue transcribing. Remember that the RNA polymerase paused right here. And now after the ribosome is caught up, it's caught up to the stop codon. Now it's going to continue transcribing. So when that happens, it's going to transcribe regions 3 and 4 right here. When regions 3 and 4 are transcribed, the RNA is pretty much going to fold in itself. Mind you, the conformation that you're going to get is this conformation on the left. So regions 3 and 4 are going to form the hairpin loop, not regions 2 and 3. You will not get this conformation because remember that your ribosome has stopped right here. It is stopped pretty much on region 2. If the ribosome is paused on region 2 cannot form a hairpin with region 3. So this over here cannot happen. Why? Because the ribosome is stuck right here. It's just sitting here on region 2. So the only conformation that you could get is this conformation. If regions 3 and 4 form a hair loop, that will yank the rest of the DNA out of the RNA polymerase and it will stop transcription. And that is how you are left with a short mRNA. Because 3 and 4 attenuated, because they formed that hair loop, it literally ripped the DNA out of the RNA polymerase. So the RNA polymerase is just like free floating again. It's not bound to anything, so it's not transcribing anything. Which means that you were left with a pretty premature mRNA. You were left with a short mRNA. And that is the second level of regulation, how the cell regulates. Do I need to make more structural genes or do I not need to? So any questions about this so far?
I just have a clarification question.
Yeah. Okay.
So we don't need to worry about the levels of tRNA tryptophan unless tryptophan itself is low, right?
That's a very good question. Damn, I actually had a problem that's very similar to that. But to answer your question, yes, that's a good point. So remember, all of this that we're talking about right now, none of this would be happening. All this attenuation and whatever, none of this would be happening if tryptophan was high. If tryptophan was in high concentration, then the repressor would be connected and the RNA polymerase can't even bind to the promoter site. So none of this would be happening if tryptophan concentration was high. That's a good point. That's a good question. Okay, does that make sense to everybody?
Yes, thank you.
Sweet. Okay, so this is the situation if tryptophan is low and tRNA tryptophan is high. So now let's talk about if tryptophan is low and tRNA tryptophan is also low. Okay, so just like here, just like before, the RNA polymerase is going to bind somewhere here and it's going to start transcribing. It's going to go la, 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 all the way until it reaches the transcription pause site. So now, once again, RNA polymerase is paused right here, like region 2. It's paused there so that the ribosome could catch up to what's been transcribed. So it's going to go la, la, la until it reaches here. Remember, in the previous situation, when tRNA was in high concentration, when tRNA tryptophan was in high concentration, translation was able to proceed. It was able to proceed because you have enough tRNA proteins to let it continue translating. But in this situation, tRNA tryptophan is low. So translation has to stop because there is not enough tRNA tryptophan to continue translation. So instead of the ribosome pausing here at the stop codon, it has prematurely paused here at the trip codons. It's paused here because it literally does not have the transfer RNA necessary to continue translating. So does that make sense so far? Okay, this is important because remember in the previous slide when when tRNA tryptophan was high, the ribosome paused in region 2. It paused right here at the stop codon, which is region 2. Now, because tRNA tryptophan is low, the ribosome has stopped in region 1. So that's the difference. Here, when tRNA tryptophan is high, it pauses in region 2. When tRNA tryptophan is low, it pauses in region 1. And here's why this is significant. So if the ribosome pauses in region 1, the RNA polymerase is going to continue transcribing. It's going to go la, 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 and it's going to make regions 3 and 4. However, now region 2, because there's no ribosome sitting on it, now region 2 is available to hairpin with region 3. And if that happens, you're going to get this confirmation. 2 is 2 because region 2 does not have a giant ribosome sitting on it, it's able to form a hairpin loop with 3. And when you get this confirmation, transcription has still paused, but it's only paused until the ribosome gets its tRNA tryptophan. So once the ribosome gets its tRNA tryptophan, it could satisfy these codons, it could place these 2 codons, and it could continue translating. It will destroy this hairpin loop, and you'll come out with a full mRNA transcript. Okay, so any questions so far? How do you guys feel about this? How did you guys feel about this before? How do you feel about it now? Are you still confused? Does this make more sense?
I feel like that this explanation was honestly it helped clear up a lot from the last study session that I had on Wednesday.
Okay, yeah. Listen, like this took me like 1,000 hours to learn. It's not easy stuff. So once you like conceptualize it, or once you practice these concepts, it definitely gets a little bit easier. Okay, so do you guys have any more questions, or do you want to move on to practice problems?
We can try practice problems.
Sweet. Okay, so I have some problems for you. Okay, let's say region 4 is deleted. What will be the size of the mRNA transcript? So what if this region right here, what if it was deleted? What will be the size of the transcript? What do you guys think? Thank you. Thanks. Oh, thank you. Okay, any takers for this one?
Wouldn't it depend on the levels of the tRNA tryptophan?
Why would you say that?
Because depending on the levels of tRNA tryptophan, that will determine if we are going to have attenuation happen or
Yes.
not.
Yeah. So I'm so sorry. I'm just a little distracted right now. Yes, you're certainly right that the level of tRNA tryptophan will determine if attenuation will occur. So let's check this out. Let's say that attenuation, or excuse me, let's say that tRNA is low, right? If tRNA is low, you're going to get this confirmation. If region 4 has been deleted, nothing would really happen, right? Because the only thing doing the hairpin whatever is region 2 and 3. These are the only regions like really having any business going on. So if tryptophan levels are low and you've deleted region 4, nothing is really going to happen. But let's say you delete region 4 and tryptophan and tRNA tryptophan concentrations are high. So remember, if tRNA tryptophan is high, the confirmation you're going to get is this one. the ribosome is going to be sitting right here on the stop codon, which is in region 2. The ribosome is going to be, once again, sitting right here in region 2, which means that region 2 cannot form a hairpin with region 3. This conformation cannot happen because the ribosome is here on the stop codon on region 2. So what would happen is 3 and 4 will attenuate and you'll be left with a short mRNA. However, you just deleted region 4. So if you delete region 4, region 3 doesn't have anything to form a hairpin with, right? It can't connect to anything. So if you delete region 4, this conformation will never happen, which means that eventually the ribosome will continue and it will make the full length protein. In either situation, in either scenario, you will be left with the complete, you'll be left with the complete mRNA transcript. Does that make sense?
Yeah, that makes sense.
And I honestly really encourage you guys to just practice questions like these. Laura loves questions like these. Ask yourself, okay, like what if region 1 got deleted? What if region 3 got deleted? You know, just practice like these little scenarios in your head and see what you get. Okay, so again, just for the recording, the answer to the first one, if region 4 is deleted, what will be the size of the mRNA transcript and why? The size of the mRNA transcript will be complete. You'll be left with the full mRNA because if region 4 is deleted, if you have too little tRNA, you'll get this concentration and region 4 doesn't do anything, so nobody cares. If you have too much tRNA tryptophan, this is the conformation you'll get. But because you've deleted region 4, 3 can't attenuate with anything. Region 3 can't attenuate with anything, so the ribosome is going to continue doing its thing. The RNA polymerase is going to continue transcribing, and the ribosome will continue translating. Any questions about that? Okay, so on to the second one. If the UGG sequence, by the way, this is the tryptophan sequence. If the which is right here. So if the tryptophan sequence is mutated to UGU, which codes for cysteine, and there is no tRNA cysteine around, what will be the size of the mRNA? And why do you think? So again, instead of so if you mutate the cell to where this tryptophan codon changes to a cysteine, what do you think will be the size of the mRNA? And if there's no tRNA cysteine around, what do you guys think?
I think that the ribosome will be forced to stop because there's no tRNA trip available.
Perfect. Yes. If there's no tRNA cysteine available, the same thing is pretty much going to happen if there's no tRNA tryptophan available. The ribosome is going to stop right here. It's going to find the cysteine codon, and it's going to be like, oh man, I don't have any tRNA cysteine. I have to stop right here. Just like if there was no tRNA tryptophan, the ribosome will get to here. It'll be like, I don't have any tRNA tryptophan, so I have to stop. And eventually you will get your full size of the mRNA, because what conformation will you get if the ribosome stops here? Will you get this conformation, the one on the left or the one on the right?
The one on the right.
Yeah, right.
Exactly. This is the conformation you'll get. Because once again, the ribosome is sitting on region 1, which allows regions 2 and 3 to attenuate, and that will enable translation later of the full mRNA transcript. Okay, so any questions about either of these questions so far? That's hot. Okay, so true or false? Attenuation will only occur when tryptophan levels are high. What do you guys think?
True.
True.
Interesting. So this question actually got me too. The answer to this question is false. Because remember, attenuation will occur when tRNA tryptophan is high. When tRNA tryptophan is high, in high concentration, you're going to get this conformation and attenuation will occur. The full mRNA transcript will not be transcribed, and evidently your ribosome can't translate everything because nothing was transcribed. But if your tryptophan levels are high, then we go back to the first level of regulation. If your tryptophan levels are high, the repressor is going to be bound here to the operator. The repressor is going to be bound to the operon, and your RNA polymerase won't even be able to bind to the DNA. So if tryptophan levels are high, you won't even get the situation. It won't even give you the chance. Like you were saying before, someone asked, would this only be relevant if tryptophan, sorry, I don't remember exactly what you asked, but your question was pretty much essentially this. So like the question was before, this is false. Attenuation will not occur if tryptophan levels are high, because if tryptophan levels are high, the RNA polymerase won't even bind to the operon at all. Does that make sense?
Yeah.
Sweet. Okay, so any questions about RNA polymerase, the different transcription factors, operons, attenuation? How do you guys
Could
feel?
you repeat the answer to question 1 again?
Yeah, absolutely. If region 4 is deleted, what will be the size of the mRNA transcript? So let's look at region 4. Let's get rid of region 4. Let's pretend it's not even here. So if tryptophan levels are high, if tRNA tryptophan levels are high, you're going to get this conformation. Do you agree?
Yeah.
Awesome. But if you delete region 4, if all of this is like deleted, then you obviously can't get this hairpin loop because there's no region 4, which means that 3 can't attenuate anything, which means that the RNA polymerase will be able to continue transcribing the rest of the DNA. It'll be able to, remember, we're talking about like the region right here. RNA polymerase will be able to keep going, transcribe all of your structural genes. The ribosome directly behind it will also be able to follow and transcribe the full mRNA transcript. So if region 4 is deleted, it doesn't matter if you have high or low tRNA tryptophan levels. You're still going to get the full mRNA transcript, and the ribosome would theoretically translate the whole mRNA transcript. Does that help?
Yeah.
Okay. Any more questions? Okay, cool. So Saeed is going to take it on from here. Saeed, you said you wanted me to continue flipping through the slides?
Yeah, if you could, I'll just say like next slide or something like that.
Yeah, sure, no problem.
Give me 1 second. I'm gonna email Jessica the slides. The PDF. Okay. All right. Okay, guys. So can you all hear me? Can you all hear me fine?
I can hear you.
Yeah.
Okay, perfect. All right. So we're just gonna go through firstly the non-coding RNAs, different types. So basically anything that's not an mRNA. And then we're going to go through RNAi and RNA inactivation, which just covers the mechanisms and pathways of mRNA and siRNA.
Actually, quick question.
Yeah.
So I asked this in the lecture, but I don't think Laura actually got to it. In the lectures that she posted on Moodle, do we have to know like all the different types of non-coding RNAs that she listed, like the transfer RNA, ribosomal RNA, PB interacting RNA, all that?
Definitely not. No, like Saeed will talk about the important ones, but you do not need to memorize every single non-coding RNA.
Okay.
Yeah, I think in that whole list, she basically just know like the basic functions of each. And I know she mentioned pRNAs, and then she like right after said that we won't like
talk about
really
those.
do it in this class. So really, like you just need to know the basics of each.
Okay, thanks.
All right, perfect. Okay, so let's get started. So yeah, basically non-coding RNAs, anything that isn't an mRNA is a non-coding one. And these are really useful and versatile for the cell because they can do a variety of things. They can do anything from catalyzing reactions when they form complexes with other proteins. And they can also repress and regulate mRNA as in RNAi. So basically they can attach to mRNA and pretty much block tRNA from forming proteins with that, and that's really important for regulation, obviously. And also another thing about RNAs is that they can come either single-stranded or double-stranded, and if they're double-stranded, then obviously they're more stable, and then there's more the cell can do with that. And we'll get to that when we talk about risk and how that basically unwinds double-stranded RNAs and then like uses one for a guide strand and such like that. And RNAs can also base pair with themselves or other DNA, and that's obviously really useful for regulation when it does base with mRNAs. And yeah, so next slide. I'll just quickly go through each different type of non-coding. So firstly, we have rRNAs, so that's just your ribosomal RNA. And rRNAs themselves are a complex of proteins and RNA, and the RNA portion of that is what catalyzes peptide bond formation. And that's basically how you get your proteins to synthesize, right? You just have the mRNA go through, and the tRNA comes in, and it just complements with each other. And based on that order of the mRNA, you'll have your correct amino acids, and then therefore your proper protein will be synthesized. And then we'll have tRNAs.
Do you want me to change slides?
Oh yeah, thank you. So yeah, and then you have your tRNAs, and these are basically just complementary bases for mRNAs, and like I said, they just carry over your amino acids to the ribosome for translation, and that's pretty much all you need to know for that. And then we have other, oh yeah, next slide.
Mhm.
And then just like other notable RNAs, we have small nuclear RNAs, and these are just like rRNAs, they're just components of spliceosomes, which the RNA portion of the spliceosome is what joins exons together when the introns are spliced out, and that just basically helps formate the formation of mature RNAs or mature mRNAs. And then you have your long coding RNAs, and these are basically, at least in humans, they're really only used for X inactivation. Basically, if you have any animal that has, the XX or XY type of genome, obviously you can't have both XXs in a cell, so one just gets inactivated. And the way that does is that you have these two long coding RNAs that complement each other, and they basically come together. And at least for X inactivation, you have T6 and Xs, which if you notice, they're just literally the names are just the verses of each other. So they just come over and they just cap the R chromosome to inactivate it. You really don't need to know that much in detail, but that's what it's used for. And then you have CRISPR, and CRISPR is basically the siRNA version that you have in bacteria, right? Si is only in mammals or only in eukaryotes. Bacteria have CRISPR, and that's basically just all it is that bacteria are able to store viral DNA. So when they do get infected with viruses, they can just transcribe that and make a guide RNA to then regulate any viral RNA and just basically target that for degradation. I think that's all for that. And then for next, we have some other smaller RNAs to be noted. Well, not smaller, but there's others. You have your SRP and your RNase, and these are both ribonucleic proteins, so they're just complexes of, again, just a ribose ribose and nucleoprotein, nucleus and protein. So yeah, anyways, SRP, you saw that for protein trafficking. These are basically how the protein gets trafficked into the RNA, and it's just made up of RNA and protein. And then you have your RNase. Again, just another complex of RNA and protein. These are just used for splicing out or cutting pieces of RNA. And the example she gave was they're used for processing tRNA. So basically, when you have your tRNA to be matured, you just cut off the pieces of RNA using RNases, and then there you go. You have your mature tRNA. And then next slide for after that. And then, so here's like the main part for this RNAi, RNA inactivation. This obviously is what you use your last two non-coding RNAs, your miRNA and then your siRNA. And these are both just forms of post-transcriptional control of mRNA. They're only found across all types of eukaryotic cells. Again, bacteria have CRISPR. And RNA inactivation is a great way for the cell to protect itself from both external, exogenous and endogenous harm. So basically, if it gets infected with a virus, or if it has, say, its own RNA that it wants to regulate, or say it doesn't need it right now, it can just use that to repress it. And another thing to know that RNAi is a very popular tool in research for protein knockdown, which we'll get to later. And so on to the next slide. First, we'll just go through mRNA. And I feel like an easy way to remember which is which, if you think of M in mRNA as in like myself or me, you can just think of that and remind yourself, oh, so this is what a cell will use if it wants to repress endogenous mRNA. So basically, this is the same diagram that she used, and I'll just go through the different steps. But an important thing I want to, I feel like it's helpful when you go through like any pathways in this class, always like think of the end goal or like the last step. So what the cell wants to do, and then kind of like work your way back from that. So for example, in this situation, you're going to have a piece of mRNA that's from the cell that's going to be transcribing when you don't, or translating when you don't need it to. So basically, you're trying to figure out a way. So how can the cell block that mRNA from matching with its tRNA? So if you go to the next slide, we'll just go through the different steps. And an important thing to note, steps 1 through 3 are all going to be inside the nucleus. Everything after that is pretty much going to be just as similar as it is in siRNA, and it all happens in the cytoplasm. The only thing, again, for mRNA, because it's from the cell's own genome, you're going to need, of course, the cell to make its own miRNA. So first we have RNA polymerase II, and that just transcribes the PRI miRNA. And also, again, like a very important thing, in that chart that she gave, there's a typo. It says pre-mRNA, but it really should be PRI. Pre-mRNA comes later in the pathway. But yeah, just make sure that that's like a very easy thing to mess up. So on to step 2, you have your PRI mRNA, and that still inside the nucleus gets cleaved by TROSHA into then pre-mRNA. And so the difference between PRI and pre-mRNA is just literally, the first is just much longer and has a hairpin loop. TROSHA just comes in and cuts that up into smaller pieces that still have their hairpin loops. They're just literally the same thing, but they're just a little smaller. And then you have your so step 3, you have your pre-miRNA. And these are carried or used, these go outside of the nucleus by using exportin 5. And then when they're outside in the cytoplasm, that's when Dicer comes in, and it just processes it even more into just miRNA duplexes. And that just means they're double-stranded. So everything up until this point is all double-stranded RNA, and everything until the dicer has a hairpin loop. So all dicer does is just comes in and it just cuts off that hairpin loop, and then you have your step 4 miRNAs. These then again are double-stranded. They come together with the RNA protein and they form the RISC complex. And basically, when that happens, RISC is going to come in and it's going to unwind the mRNA. And it's going to decide, all right, I'm going to pick one strand to use as a guide strand, and the other just gets degraded. And whatever the guide strand is, that is then used step 5 to be a complement to target mRNA. And that basically just marks it for degradation. And an important thing, the reason I starred complementary, is that in humans, most of our miRNA is actually not 100% complementary. And that's important for 2 reasons. And so basically, for the reason that's important is that if your miRNA isn't 100% complementary, if it's just good enough, then I feel like a lot of things in biology, if it's just good enough, then you can use it for multiple different other things. So if the miRNA is almost an exact match for the mRNA, then it can be used for multiple different mRNAs in the same pathway. And basically that's And the benefit of that is just that it can, in one pathway, that way you can pick multiple points within that same pathway to knock down and slow down translation. So you could use the same miRNA. The cell doesn't have to make too many. You can just make the one, and then it can, in multiple different parts of the same pathway, regulate the transcription. And the second point why it's important is that if your miRNA isn't 100% a great, perfect match for the target mRNA, then it's not degraded per se. It's actually sequestered into a P-body. And if it's in a P-body, it's just used there either for storage until later when like the cell does have use for the mRNA, or eventually it does get degraded. Or the other thing that can happen is that the ribosome will just decrease its translation of the mRNA. So again, it doesn't get degraded straight away. It gets sequestered into a P-body for later use, or it just decreases the ribosome translating the mRNA. Any questions for this pathway so far? If not, then we can just go to siRNA. All right. Okay, yeah, so this is just, again, the same diagram that she gave us. Basically, siRNA is a lot, in a sense, it's a lot simpler. You just cut out the beginning part of mRNA because literally what the cell does in this case, if it gets infected, say, with a virus, it just uses its own viral DNA against it. So basically, it has like most of the work cut out for it. So if you go to the next slide, we can go through the steps. Basically, it'll take the double-stranded or the short hairpin RNA from the virus, and it'll just, again, dicer will come in and it'll just cleave that. It'll just snip off the hairpin loop, and then you have your siRNA duplex, so just the double-stranded, and that'll just again make it'll just come together with arginine protein to make the RISC compound or complex, and then RISC will then proceed to unwind the siRNA duplex, and again pick a guide strand and then one strand to get degraded, and then that guide strand will then form a complex with the viral mRNA, and then that way the mRNA gets spliced out and then gets degraded by arginine. And the only really important thing to note here is that because the cell is using the virus's DNA again or RNA against itself, it's going to be 100% complete match. So that's the big difference between the two pathways. For siRNA, it's only going to get degraded, and that makes sense because if it's a viral DNA, then the cell doesn't really need it to be sequestered in a P-body, and it doesn't have the ribosomes to slow its or repress its translation.
Quick question.
Yeah, go for it.
So for step 2, you said that siRNA duplexes is unwound by RISC, right? And it's double strand at that point, correct?
Yeah.
So when RISC unwinds it, does that mean that it changes to single strand?
Yeah, the way I understand it is that it's the same at that point as it happens in mRNA, so it just unwinds it. And yeah, it'll be single stranded after one half gets marked for a guide strand, and the other part just gets degraded.
Okay, thank you.
Yeah. So yeah, that's pretty much the si pathway. And then if we go to the next slide, we'll just have like, oh, you can go to the next slide. Oh no, one.
Sorry, did I skip one?
Yeah, thank you.
Oh, this one?
Yeah.
Okay.
Okay, so, and then just to like wrap it all up, just like important differences and similarities between the two pathways, really there's a lot more differences. So for the differences, everything before the RNA duplex enters the cytoplasm and makes that RISC complex, that's different because again, for mRNA, you're gonna have the cell actually has to make the RNA and actually has to export it from the nucleus, whereas in siRNA, the RNA is already provided for it by the virus itself. And in humans, the mRNA is not 100% accurate, but that's just so it can be used for multiple different mRNAs in the same pathway. and siRNA is the one that researchers use in cell therapies and protein knockdowns. So because in siRNA it's 100% match, what you can do in a lab is that you can use it to knock down protein. So, right, what you can do is you can go in a cell and you can insert a piece of DNA, and then you can see if I knock down this part of a pathway, or if I take out this protein, what's that going to affect, right? Or what's that like loss of function going to be? And with that, you can basically work your way back and be like, all right, so if I do that, and then the cell can't do this, so that must be the function of whatever protein that I knocked down. And that's obviously very useful in cell therapies, and just useful like for understanding the cell mechanisms in general. And then for similarities, basically the whole part with Dicer and RISC is going to be the same. You're going to have in both cases you're going to have a double-stranded RNA that's going to get unwound, and a RISC is going to pick one to be a guide strand to target the mRNA, and then one to degrade. So yeah, and then we'll have some practice problems. If you guys don't have any questions up until this point. No, okay. So question number 1, what's the advantage of miRNA not being 100% complement to mRNA in humans, and why isn't this necessarily the case in siRNA?
It's the advantage because it can be used in multiple pathways to slow down RNA.
Yeah. And why wouldn't you want that in siRNA?
I forgot.
No, it's all right. So yeah, that's the second piece of, have you got the first piece right, that you can use the mRNA to like target multiple points in the same pathway. But also what happens when the mRNA isn't a 100% match is that the cell's not going to degrade the mRNA. It's just going to sequester it into a P-body for later use. Right? Because cells want to be very efficient. They're not going to want to use its own DNA or its own mRNA when it doesn't need to. So it'll just sequester that. It'll just put it away in a P-body for later use, or it's just going to repress the ribosome for translation. And the reason why you don't really need that in siRNA is because, again, it's going to be an exogenous source. So you wouldn't have the ribosome to make it in the first place, and you wouldn't need to sequester it either because the cell's never going to need to use a viral piece of DNA.
Hey, Saeed, if I could interject real quick, there's a question in the comment. What's the difference between silencing and degrading? Or
degrading?
Are they
That's a
like
good question. I feel like silencing is just going to be a more like broader term. Again, like sequestering a piece of DNA or an mRNA, so silencing can also be degradation, but it can also include stuff like sequestering and storage or repressing translation. But degradation is just what it is. You're just going to be getting rid of the piece of RNA. You're not going to be able to use it later.
Got it.
If that's.
Does that help the people asking the question?
Yeah, it does. Thank you.
No problem.
Okay. All right, so we can go to question number 2.
Could you go through question number 1, part 2 again?
Oh yeah, sure, of course. So do you mean the part about siRNA?
Yes.
Yeah. So basically, if you want to think about it, two things happen when the mRNA isn't 100% match to its target mRNA. The first thing is that since it's not 100% match, it can be used for the same piece of mRNA can be used for multiple different RNAs, because again, like, it has to be complementary. But if it's just good enough, then the cell can use it for different mRNAs in the same pathway, because they're all to a certain degree going to be similar to each other. And then the second thing that happens is that if the mRNA isn't a complete match to the mRNA, then the cell's not going to just mark it for degradation like it does in siRNA. It's going to either sequester it to a P-body for later use, or it's just going to turn down the ribosome that's making the mRNA. So if that answers your question.
Yeah, so why isn't this case in siRNA?
Right. So again, like for siRNA, you're gonna have the cell targeting a piece of RNA that it doesn't produce. It's gonna be from outside. It's gonna be like from a virus, for example. So if that's the case, then the cell doesn't have any use for sequestering it to a P-body, and it doesn't have any use for, and it's not gonna have the ribosomes for it either. So it can't, turn down its translation. And also because it's using a piece of the virus's RNA against itself, it's going to be 100% matched since it's the same virus that's going to be, targeting its own mRNA.
Okay, that makes sense. Thank you.
Yeah, so basically mRNA comes from the cell's own genome and siRNA comes from any other like foreign invader. Okay, so number 2, what's the difference in structure between PRI mRNA, pre-miRNA, and then the mRNA duplex, and what causes those changes, or what processes to those different stages? Okay, I'll just go through. So basically, pre-PRI mRNA is the first step, and the only difference is that it's just literally much longer than pre-miRNA. And what happens is that drosha comes inside the nucleus, and it just cleaves PRI miRNA into smaller pieces. Because remember, it has to get exported out of the nucleus, so it's going to want to be in a smaller size. But both the pre and the PRI mRNAs are going to have that hairpin loop, which Dicer comes in and then cleaves off the pre-miRNA in the cytoplasm. And after it cleaves off the hairpin loop, then you're just left with the mRNA duplex, which again, duplex just means double-stranded in this case. And yeah, that's about it. If you guys have any questions about anything we went over.
For the PRI mRNA, you mentioned that like Drosha's cleaving the PRI mRNA hairpin, and then Dicer's cleaving the PRE mRNA hairpin.
No. The hairpin's gonna be there after Drosha does its processing. Drosha just literally, you can just think of it as cutting the mRNA into smaller pieces. It leaves the hairpin and all that. It just, because the PRI mRNA is literally just a much longer base pair than what the cell can export. So Drosha its just job is to cut it into smaller pieces, and then Dicer's job is to cut off that hairpin loop.
So Dicer is working in both PRI and PRE mRNA?
No. Dicer is only in the cytoplasm. So when the cell does cut up the PRI into the smaller pieces, then it can export it through exportin 5. And then when it's in the cytoplasm, that's where Dicer is. And then it can cut off those hairpin loops.
Okay. So
Drosha,
is PRI and PRE both in the nucleus?
yeah. PRI is just too long. Pre is the smaller version of it that Drosha cuts up, and then that goes into the cytoplasm where Dicer can get it.
Because I thought it was like PRI got cut by Drosha, which then forms PRE.
Mm-hmm.
And then PRE goes into like the cytosol.
Yeah, that's exactly
right. By Dicer.
Yeah.
Okay.
In the slide she gave, the first pre-mRNA is actually PRI. It's just like a typo.
Mm-hmm.
So first you have the I, and then it gets Drosha processes it into the pre, which then gets exported out, where Dicer can cut off those hairpin loops.
Okay.
It's very confusing, but I feel like the best way is just to like draw it out or like write it out, and also like talk it out to maybe a friend or something.
Mm-hmm.
Because it's a lot of small steps that are very easy to interchange, and that's really like a lot of the different pathways that we're going to go over in this class.
Okay, so the PRE is really just the smaller pieces of cut up PRI mRNA.
Yeah. Yeah.
And then the PRE will get exported out, and then Dicer will cut it into even smaller pieces, forming the duplexes.
Yeah, it's gonna cut it to smaller pieces and it's gonna cut off that hairpin loop. And then you have your duplex that can form the Arisk complex.
Okay, that makes
So
more.
basically, think of it as first you need to get into smaller pieces, then you need to take off the hairpin loop, and then you need to unwind the double strand.
Okay, that makes more sense. Thank you for clarifying that.
Yeah, no problem. Yeah, if you have any other questions.
Do you like go through problem set question 2? Why I feel like the A part and B part they basically ask the same question.
Yeah, sure, we can pull up problem set. You're asking for question 2, right?
Yes.
Okay, let's see.
Can you say what that question is?
Is it the one experiment with Milo and Fry?
Yes, that's the
Okay.
one.
And are you what are you asking again? Sorry.
I feel like the part A and part B they are asked the same question. One is why double strand RNA works and why single strand RNA doesn't work. I just like, yeah.
Yeah.
Oh, okay, wait, I see what you're asking. Why sense RNA will not work, and then why would double stranded be more effective? Okay, yeah. And Sebastian, if you think this is wrong, feel free to but I think it's more asking about in a sense it's asking about stability and right, because if you have one's definitely going to be more stable than the other, and if one's double, then it can be used for more things than a single. Sebastian, does that sound right?
Yeah. Okay. Sorry. Let me just open that document. Can you guys see the screen? Let me just open the problem set just so I know what I'm like looking at.
Also, while we're at it, can we also do you think we can go for part A a bit more in depth? Because I honestly really do not understand what's asking.
Sure. Sure. Let's go through this one by one. So for problem 2, the first one says, explain why sense RNA probably would not work. Sense RNA was what? Sense RNA was the single-stranded?
Oh, right. Okay, so that's the single-stranded that's identical to the target mRNA. And the antisense
Yes.
is complementary.
Yes.
Okay. So if you think about how it works, right? So if you think about how translation works, right, you're gonna want an mRNA and then a complementary tRNA. But in this case, you have a single-strand RNA that's identical to the target. So why wouldn't that work? Right, because if you have the way normally it works is that you have the one mRNA strand that forms a complementary match with a tRNA, or if it was an RNA inactivation, it's going to form a complementary bond with an miRNA. But if you have one that's identical, you can see why that wouldn't. If that explains it.
Can you explain that again, please?
Um,
I have a question. You said it has to be complementary to the mRNA or
yeah. If you think about how base pairing works, in general, right? You're gonna have a G that matches with a C, and a U that matches the T, right? You're gonna want to have bases that are complements to each other. You can't have bases that are the same, right? You couldn't have a G match with a G, for example. If that clarifies a little bit better.
So the guys be complementary?
Yeah, you couldn't have one that's identical, right?
Okay, but so why would that be the case?
Oh, in the sense, right? Because it gives you in the question that the sense RNA, by definition, is a single-stranded one, and it has an identical sequence to that target mRNA. Right, whereas an antisense would be one that's just complementary, and a double-stranded would be the sense and the antisense, right? Because if you think about it, when you do have a double-stranded RNA, that's where risk comes in and unwinds, and it picks one of the strands, one of the sense or the antisense to be the guide, right? It couldn't pick the sense to be the guide because they wouldn't come together if they're identical. It would have to pick the one that could be used as a guide, right? It'd have to pick the one that's a complementary. If
Yeah.
that's if
Sorry, continue.
no, that's all I have I don't know if that clarifies it. It is confusing, but
Yeah, essentially the differences between part A and B of question 2 is question 2, part A says, why does the sense RNA, why does the single strand sense RNA not work? And like Saeed said, there's an identical sequence and there's a complementary sequence. So if you're using the identical sequence, part A is asking you to describe or to explain why does the identical sequence not work. Part B of question 2 is saying, why does double-stranded RNA work as opposed to single-stranded RNA? And just a disclaimer, nobody really knows why RISC has a strong affinity to double-stranded RNA as opposed to single-stranded. But think about, you can propose some reasons why. So you could think about the stability. So what's the difference in stability between double-stranded RNA versus single-stranded RNA? You could think about like the KD relationship between RISC and the double strand versus RISC and the single strand. Or what was the other one that I was just telling someone? Yeah, so RNA stability. What is RISC's purpose? Like, what is RISC? What does that protein want to do? What is it like doing? And what is it used for? If that makes sense. Does that clarify your question?
I think.
it's hard because like
It's a little
I
tricky.
feel like it's very hard to like not give out the exact answer, but if that helps you, I feel like that's a good like hint towards the right.
I think that should be fine. I think I probably might have to go back and watch the lecture to understand it.
Sure. And of course, you're welcome to email TAs or go to their office hours.
Yeah, definitely.
Or Laura's, yeah.
Can I ask a question on it was on the problem set. It was like part of question 1.
Sure.
It was like the double parter where it had the stop codons mutate, or the TRP codons mutated into stop codons. That one.
Yeah.
So I was answering the question assuming that the ribosome is just going to be like stuck or like paused on it. Is that the right way about going about it, or is the ribosome going to dissociate from?
Yes.
So I won't like say how I guess.
Yeah, I'm struggling to like answer your question without revealing the answer, but essentially,
that's like the hard.
Yeah. Your explanation sounds right.
I'm kind of like, I'm wording it as if like I kind of explained it as if there was like a low level of TRP charged tRNA, like because the ribosome is still going to pause on there. So it's like the ribosome is experiencing, or the whole like complex is experiencing like a low tRNA or a TRP charged tRNA, like, but instead of there being low tRNA or tryptophan, there's just the stop codon is what's stopping it. That's how I was going about it, if that makes sense.
Yeah, so I can't confirm or deny your answer, but you're on the right track.
Okay. And I think that was
okay. Do you guys have any more questions about anything we covered in the review?
I have a question.
Yeah.
I have a question about the leader sequence, the region. I'm still a bit confused because So does the leader sequence have more to do with the trip or the trip tRNA?
I'm sorry. Could you repeat your question?
Sure. Does the leader sequence, does it have more to do with the tryptophan or the trip tRNA levels?
Good question. The leader sequence. So pretty much everything that we're looking. Can you see this diagram?
Yes.
Yeah. So everything in this diagram, like all of these regions, 1, 2, 3, 4, star codon, whatever, all of this is just the leader sequence. This whole diagram is just this stretch right here. So yes, the leader sequence is more concerned with tRNA trip concentrations than the overall operon is. In the overall operon, it's just asking, is there enough tryptophan? Yes or no. And this one, it's asking, is there enough tRNA tryptophan? Yes or no.
Oh, okay. So because I'm looking at the problem set and there's a question on there that has to do with it. That's what I'm wondering.
Yeah. Okay.
Okay. Okay, thank you.
No problem. All right. Do you guys have any more questions or are you all set?
Does this count as check-in? If
No.
no, okay.
No. You're checking. I mean, you're David, right?
Yeah.
I got your email, so I could check you in. But just for future reference, I don't think reviews count as check-in. You'd either have to email the TA, email Laura, or go to help sessions.
All right. All right. Thank you.
No problem.
Yeah.
All right. See you guys. My guy. Let me stop the recording.
Oh, yeah, you're definitely